Hello and welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Sarah Paramore and I'm a postdoc at the University of Chicago and also the SDB postdoc representative. And I will be moderating today with Zong-Yuan Liu, a postdoc at the University of Michigan. We are excited to highlight the work of our outstanding postdoctoral members. Today, Gopal Kushawah from the Stowers Institute for Medical Research and Neha Ahuja from the University of Texas Southwestern Medical Center will be sharing their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please enter any questions that you have in the Zoom Q&A box. So I am delighted to introduce our first speaker, Dr. Gopal Kushawah, who is currently a postdoctoral researcher in Ariel Bazzini's lab. Gopal received his bachelor's degree from Ambedkar University, where he studied zoology, botany, and chemistry. He went on to do a master's in biotechnology at Georgia University before doing his doctoral work at the Center for Cellular and Molecular Biology in Hyderabad, India. As a postdoc, he has developed strategies to use CRISPR for efficient gene knockdown in vertebrate embryos, including the zebrafish, which you can read about in some of his published work. But today we are excited to be here to hear about what he has been doing more recently, investigating the spatiotemporal roles of maternal mRNA in the developing zebrafish embryo. So please welcome Gopal.
Thank you, Sarah, for nice introduction. Today I will talk about one of my story where I am looking for the function of spatiotemporal maternal RNA during early embryonic development. One of such gene we identified CTH1, which is working as a maternal RNA decay factor, and it is essential for zebrafish development. In eukaryotes, female deposits a large fraction of RNA even before fertilization, and this RNA later is known as maternal RNA, which is present predominantly for a short period of time, and it constitutes 1/3 of the zygotic genome. And this maternal RNA degradation is required for zygotic genome activation. And during this process, the control shifts from the maternal side to the embryonic side. So in short, the maternal RNA decay is very crucial for embryonic development. And that's why people have studied maternal RNA decay in different organisms. But due to technical limitation, almost all these studies, they were kind of focusing the whole embryo as a single entity, and they do not consider the embryo as a multicellular during development. So they were not having the spatial context in their studies. Here we hypothesize that during development, these embryos become multicellular, and due to that, there should be certain maternal RNA which gets degraded in a spatiotemporal manner, and then they regulate embryonic development post-transcriptionally. So if this is true, then questions arises how to identify such maternal RNA and whether they have some functional significance or not. So to do that, first we asked how to identify these RNA. So for that, our lab did SLAM-seq. It's a technique where we use chemically modified uridine and we inject them in by microinjection in zebrafish embryo at one cell stage. So if there is new transcription event, this chemically modified uridine will go and gets incorporated into newly transcribed RNA. So if there are any RNA which do not show its incorporation, basically they are maternal RNA. So we selected those maternal RNA which do not show incorporation of 4-thiouridine by 4 hours, and they should be decaying with time. So now we have a list of maternal RNA which are getting degraded over time. Now question arises, what about their spatial context? So for that we utilize Satyaj's single cell RNA-seq data at mid-blastula stage, where they did single cell RNA-seq in mid-blastula zebrafish embryo, and then they combined this single cell RNA-seq data with all the known RNA in situs, and then they calculated the probability density of each RNA to present in this artificial bin of an embryo. So based on that, when we incorporated our maternal RNA data to this data, we could see that there are certain maternal RNA which are highly enriched in the marginal region as compared to the non-marginal region. So when we plotted marginal versus non-marginal genes, we could see that at the bottom, we could see some red color dots, which are basically maternal RNA. They are kind of highly enriched, suggesting that there are certain maternal RNA which are getting localized to the marginal region of an embryo. So in this study, today I am going to talk more about CTH1, but if you are interested about other candidates and whether they have some functional significance or not, you can refer to my preprint where I have discussed about other candidates as well. So coming to the CTH1, CTH1 is a gene of unknown function in zebrafish, and people have not studied it extensively. And when we did RNA-seq, and we found that CTH is among the highly deposited maternal RNA which gets degraded over time, suggesting that CTH is among the top maternally deposited RNA which is getting degraded with time. And then we looked in our RNA-seq data whether it is also getting transcribed during early embryonic stages or not. We found that in SLAM-seq also the maternal component is very highly deposited and getting degraded, while the zygotic transcript is coming around 4 hours, but the level is very low, and we see that by 6 hours, still it's very low. But the question arises, what's happening to this CTH transcript, which is zygotically transcribing at later time point? That data I will show you next slide. But before that, we wanted to know whether we still see their spatiotemporal dynamics in an embryo or not. So for that, I did SCR, and here you can see when I did SCR for endogenous CTH, you can see that it is uniformly deposited at one cell stage, and around by 2.5 hours also you can see that it is uniformly distributed, and with time it is getting degraded and getting localized to the marginal region, suggesting that yes, CTH maternal RNA is highly dynamic in nature and getting degraded and getting stabilized in the marginal region. Now coming to the question, what's happening after 6 hours, whether it is still there or not coming back. So when we looked in single cell RNA-seq data, which is up to 5 dpf, we could see that here also it is showing that it is expressing during early, it is deposited during early time points, and basically in certain cell lineages, which are basically the blastomere, and even in bulk RNA-seq data, you can see that up to 6 to 8 hours it is there, but after that up to 5 dpf in bulk RNA-seq data, also we are not able to detect it. So after next question comes how and what is controlling the decay of this CTH during early embryonic development, whether it is zygotically controlled or maternally or by both. So for that, we did another experiment where we injected alpha amanitin, which is a transcription inhibitor. So if you inhibit transcription, there should not be any zygotic factor controlling it. And still if we see its degradation, it means it is maternally controlled or maternally programmed. And that's what we saw here in alpha amanitin time course RNA-seq also. This CTH RNA is getting degraded, suggesting that CTH decay is maternally programmed. So we know now that some maternal factors are controlling it, but how it is getting localized to the marginal region. So there were studies which talks about the germ cells specific 3' UTR of certain genes which can stabilize the reporter genes or these genes in the germ cells. So this mechanism was kind of very well known for most of the germ cell genes, but is this true for other somatic cell also or not? So we wanted to test this for CTH. So we cloned CTH 3' UTR downstream to GFP and injected along with injection control. So if CTH is driving the localization to the marginal, we should see the GFP RNA localization to the margin like this. If not, we should see uniform distribution just like injection control. So when we did this, you can see here The injection control is uniformly distributed, as well as GFP with other 3' UTR is also showing uniform distribution. But when we did it for CTH 3' UTR fused with GFP, now we can see that GFP RNA is getting localized to the marginal region of the embryo, suggesting that it's the CTH 3' UTR which drives its spatiotemporal patterning. Now question comes, okay, this is GFP mRNA, whether GFP protein is also getting localized to the marginal region so that it can function locally or not. So when we look for the protein, we found yes, the protein also for GFP when it's fused with CTS 3' UTR, it's also getting localized to the marginal region, suggesting that this CTH protein is most probably functioning in the marginal region preferentially. So after this, the next question we ask, what region in this CTH3' UTR is regulating these spatiotemporal localization, whether there are only one cis regulatory element, many, or some trans-acting factors are working on it. So for that, what we did, we just based the whole 3' UTR and made a 3' UTR tiling library and then injected again them in zebrafish embryo and then collected RNA at different time points, did RNA-seq. And then what we found that as compared to means early time points, at later time points, we could see that there are certain unstable regions in these 3' UTR, suggesting that yes, there are certain fragments in this 3' UTR which is causing instability. And then when we did HCR for these three different unstable fragments, as well as one fragment which is non-responsive, we found that these unstable fragments also tend to localize in the marginal region, suggesting that these fragments are prone to instability. And interestingly, when we look for the conservation, we found that these three unstable fragments are showing a strong vertebrate conservation, especially in fishes, suggesting that these regions are having some evolutionary conserve function in these vertebrates. So after that, we have characteristics. Okay, this CTH RNA is getting degraded, and its 3' UTR is causing its localization to the marginal region. But what about the function of CTH? So for that, what we did, we generated CRISPR-Cas9 mutants for CTH using single guide RNA independently or a combination of three guide RNA, and we injected and looked for the phenotype at early time points and later time points. Here you can see that at early time point, we do not see any phenotype coming in these CTH mutants. And when these fish reaches to maturity, apparently at this time point also, we do not see any phenotype here. But when we cross these fish to generate the next progeny so that we can have maternal zygotic mutants, what we found whenever we are crossing mutant fish with their wild type siblings, either mutant female or mutant male, we are seeing embryonic lethality. And when we look closely, we found that at even early time points, we are seeing that embryos are not normal, even with wild type, in mutant female as well as mutant male. So we suspected maybe their gametogenesis is affected, or what exactly is happening. So we found that in these mutant females, the oocytes are completely degraded and they are disintegrated as compared to wild type, as well as the mature sperm from mutant males. They are also completely disintegrated and they are absolutely not looking normal. And that's why now we cannot generate the maternal zygotic mutants for CTH. So this limited us to study the function of CTH during early embryonic development in zebrafish, and this is one limitation of Cas9, that we cannot study the function of embryonic lethal genes, especially for those when their gametogenesis is affected. So here I utilize my previously published technology, which is CRISPR-Cas13, by which you can basically knock down RNA at early embryonic stages, and by that you can find their function. So when you combine these both technology, you can basically complement the limitation of Cas9, and then you can study the function of even embryonic lethal genes. So using this technology, Then what I did, I designed guide RNA against CTH and then injected them and looked for the phenotype at early time points, later time points, and also validated their knockdown. So I used three independent guide RNA. You can see very strong knockdown with three independent guide RNA. And when we look for the phenotype, we could see that as compared to wild type and controls, the phenotype in these embryos, they are developmentally delayed by 6 and 8 hours. And even at 1 dpf, dpf is day post fertilization, the body axis deformities are there with individual guide RNA, suggesting that these knockdowns are specific, but we wanted to rule out any off-target activity. So we did some rescue experiment where we provided CTH mRNA with synonymous mutations where this guide RNA cannot bind. And with that, we were able to rescue the phenotype, but in control, we could not rescue the phenotype, suggesting that these knockdowns are specific. Now we wanted to know the molecular function of this CTH. So what we did RNA-seq for two independent guide RNA, and here you can clearly see that there is a strong knockdown from two independent guide RNA. And then we wanted to find some common genes which could be the targets of CTH if they are differentially upregulated in both of these knockdowns. So we look for those genes which are kind of differentially upregulated in both the knockdowns, and then we also selected four random group of genes which are not differentially upregulated or downregulated. So from here, we then started looking what are the features in these differentially upregulated candidates, what exactly they are and how they are behaving. So we look back for these potential targets in normal RNA-seq data, and what we found when we compared their poly-A-tail status, that these potential targets, they are highly polyadenylated at early time points as compared to four randomly selected control genes. And these genes with time, they are getting deadenylated, suggesting that they are getting degraded with time due to deadenylation in wild type conditions. But in knockdown, they are kind of stabilized when CTH is not there, suggesting that CTH is causing their decay by polyadenylation. So now next question after this, we wanted to know what kind of motifs are there in these genes, and we found that their 3' UTRs are highly enriched with AU-rich sequences. And interestingly, we found that motifs which are either having 3A or 2A in between, they are highly enriched, and we found different combination of these motifs which are highly enriched in these potential targets. So after that, we wanted to check how these targets are behaving and how these different motifs are responding in wild type conditions as well as in knockdown conditions. So whenever we are having more than one site, they are responding very strongly in wild type conditions, suggesting that they are showing maximum decay as compared to genes which are having just single motif or 2A mer. But when we look this data in CTH knockdown, we found now that these all targets are stabilized, and interestingly, those genes which are having more than one target site, they are showing strong response or strong stability, suggesting that CTH is working quantitatively on these candidate genes. And even in alpha mannitine data, we found that in alpha mannitine control, they are getting degraded, suggesting that maternally controlled, but upon CTH knockdown, they are again getting stabilized, suggesting that, okay, CTH is interacting in a maternal function. So after that, we did a reporter assay to confirm that CTH is interacting with the 3' UTR of these sequences or showing causing their stability when CTH is not there. So what we did, we cloned 5 different 3' UTRs downstream to GFP and injected them in wild type conditions as well as in CTH knockdown conditions with different controls. And what we found that upon CTH knockdown, now these different 3' UTR, they are getting stabilized as compared to the control 3' UTR, suggesting that these 3' UTRs, they are responding to CTH, and upon CTH knockdown, they are getting stabilized. So after that, when we were doing these experiments and we were preparing our manuscript, there was one paper came parallelly in basically in bioarchive. They talked about RBP scan where what they did, they fused RBP RNA binding protein with ADAR, a catalytic domain, and then they provided the potential motive for these RBP to interact. So if there is any interaction, then you will see ADAR activity on this stem loop structure. So based on that, they were telling that that RBP is interacting with specific motifs or not that or not. So on that basis, they also did for CTH and they found that okay, there are AU-rich motifs in the potential targets of CTH, which are where this CTH is interacting. So when we
You
put
have
our data,
1 minute left, Gopal.
yeah, when we put our data over this, we found that yes, we are also finding these motifs. So now next question comes, if CTH is really interacting with these motifs, what if we mutate these motifs? Then we should not see the CTH, these targets response to the CTH. So we did this experiment where we mutated the CTH, potential CTH motif where it can bind, and what we found that in control, we see less GFP, but when we mutate it, now we start seeing more GFP, suggesting that motif deletion attenuates CTH targeting on these 3' UTRs. So with that, I think I was able to show you today that CTH is among one of the highly deposited maternal RNA, which rapidly gets degraded in a spatiotemporal manner and gets stabilized to the marginal region of the embryo. And this stabilization is mediated by its 3' UTR, and there are cis-regulatory elements in its 3' UTR which cause its stability. And then I also showed you that CTH is working as a RNA decay factor, which is binding to the AU-rich motifs, and then it is causing their decay. And I also showed you that Cas9 mutants are infertile for CTH, and with Cas13, you can complement this limitation to find the function of CTH during early embryonic development. And with that, I would like to acknowledge my lab and different core facilities at the Stowers Institute. Thank you. And I would be happy to take any question.
That was awesome. While we are waiting for questions in the Q&A, I was really struck by the fact that your mutants, when the Cas9 mutants have this like really severe gametogenesis defect in both the males and the females. So I was curious if you have any thoughts about like why that is like specifically the case and that the fish are fine otherwise. Like I am unfamiliar with like RNA binding proteins and stuff. So is that like a surprising result to you?
Uh, Thank you for this question. It's not surprising. The most potential answer or the probability is that since Cas9, it mutates the zygotic copies of this and CTH is not expressing or very low expressing at early time points. We are not seeing the phenotype at early time points. And later CTH starts expressing specific in certain germ cells, basically in germ cells in other cells, and that's why we are seeing those phenotypes coming in Cas9 mutants.
We have a question in the Q&A from Jayesh Sundaram, who asks, do you see AAU, AAA nuclear poly-A signal-like motif in previous MPRA slash RESA studies?
Yes, we do see.
And then we have a question from Guy who is asking, since the precise localization of CTH1 to the marginal region is accomplished by it being stabilized there, what stabilizes it there?
Yeah, since the precise local CTS to the region is by its being stabilized there, what stabilize it there? Yeah, this is very interesting question, and we are trying to find the answer for that, and we are looking for certain other factors which are there in marginal region, especially for the RNA binding proteins. But there is one potential hypothesis that CTH may be auto-regulating CTH itself in the marginal region, and this could be the negative autoregulation. So we are working currently on that hypothesis, and soon we will be having answer for that.
And then we have another question from Michael Layden. Is there a pattern to where the CTH1 targets are degraded? Is it only in the margin, or is CTH1 translated and functional throughout the process of being stabilized in the margin?
This is another very interesting question. When we look for these CTH targets, localization, we see that they are specifically not highly enriched in the marginal region, what they are present either uniformly or in the other part of the embryo. So, and when we are knocking down CTH, we start seeing their expression in the marginal region as well. So they are basically expressing in other part of the embryo.
Just give a little bit longer to see if there are any more questions in the Q&A or the chat. Okay, well, thank you so much for a great talk, and I will turn it over to our second moderator for today.
Thank you.
Thank you, Gopal, for the excellent presentation. My name is Zong-Yuan. I'm a postdoc at the University of Michigan. So it's my pleasure today to introduce our second speaker, Dr. Neha Ahuja. So Dr. Ahuja is currently a postdoc researcher at the University of Texas Southwestern Medical Center. So where her research focuses on the hyposignaling in the pancreatic development. So during her PhD at the Colorado State University, she conducted research on the biomechanical regulation of ventricular valve development in the zebrafish. So her excellent work has been recognized through several prestigious awards, including the K9I Fellowship, the GDRF Postdoctoral Fellowship, and the F32 Fellowship. So please join me in welcome Dr. Ahuja for her presentation today.
Hi, thank you so much for that very kind introduction, and thank you all for being here with me. I'm really excited to tell you about some of my work looking at myelin and its role in pancreatic development. So the pancreas is a pretty important organ. It's composed of three major cell lineages, the first of which is the endocrine cells, which contain beta cells which secrete insulin and are really important in conditions like diabetes. In addition to the beta cells, there's also the exocrine system, which consists of yellow acinar cells, shown here. These are cells that secrete digestive enzymes like amylase, and then the ductal network, which is shown here in this light blue color. And the job of the ductal cells is to connect the acinar cells to the rest of the digestive tract, so when those enzymes are secreted, they can get to where they need to go. Defects in the differentiation or organization of these lineages are really important. They are causative of things like congenital pancreatic diseases, and often predispose patients to really serious consequences like diabetes or pancreatic cancer later in life. So it's really important that we understand sort of the basic molecular mechanisms by which these organs are, or which these cell lineages are organized. So how does the pancreas develop? So our lab uses mouse model to study this question. And in mouse, the pancreas is first really apparent at about embryonic day 10.5 to 11.5. And at this stage, the pancreas consists of this sort of bag of unpolarized epithelium shown in this diagram here. The key morphogenetic process that's going to happen at this time point is a process known as de novo luminogenesis. So these cells are predominantly unpolarized at this stage. They're going to select cells within this system are going to start to acquire polarity and form these little microlumens. Those microlumens are then going to fuse into a luminal plexus. So shown here is a schematic of what the luminal plexus might look like at E14.5, just a couple days later. And at this stage, the pancreas is really a monolayer. So if we look a little bit more closely at what's going on, we'll see that at the tips, we now, instead of having a sort of stratified epithelium at 11.5, we have a single layer of cells that surround a lumen. The tips are sort of still growing, so they're going to be undergoing more de novo luminogenesis, which is a process mediated by vectorial transport. And then at the core, we've also achieved a monolayer, and this is predominantly undergoing lumen extension and remodeling. So over the last few years, it's become really abundantly clear that the Hippo pathway is really important for both pancreatic morphogenesis and cell fate. Just as a brief review, the Hippo pathway is a kinase-kinase cascade that ends up regulating these transcriptional co-activators YAP and TAZ. When the Hippo pathway is off, YAP and TAZ are free to translocate into the nucleus and affect transcription of their target genes. But when Hippo pathway is on, these kinases, LATS1 and 2, become phosphorylated, and they in turn phosphorylate YAP and TAZ, targeting them for sequestration and degradation. There's been some work on Hippo in the pancreas already. And so this is an IHC of an adult pancreas from a human. And you can see that in the ductal cells, there is really, really strikingly strong nuclear YAP. And then some of these astronef cells shown here, YAP is relatively low or cytoplasmic. There's a cell fate component of the Hippo pathway and how it is localized. And importantly, if you knock out YAP from the early pancreas, you get a really interesting effect on endocrine cells. So a knockout of YAP actually increases endocrine cell mass, even though it is eventually lethal. So we know that Hippo pathway is really, really important, but we still have a few questions about how Hippo is regulated and also what exactly YAP and TAZ are doing to facilitate morphogenesis. I became really interested when I came into the lab of this molecule called Marlin. Marlin is an upstream regulator of the Hippo pathway. It is able to interact with the Hippo pathway in a couple different ways. It's able to go directly into the nucleus and interact with YAP and TAZ there, and also interacts with some of the upstream components. Importantly, in my data, we were able to show that during lumen formation in the early pancreas, Marlin is strongly apically expressed from the earliest stages of lumen formation, and it becomes increasingly restricted to the apical membrane as time progresses. And so we thought it was sort of a good candidate to potentially link the morphogenetic events required for pancreas development with some of the YAP and TAZ transcriptional outputs that facilitate cell fate. And so that's exactly what my hypothesis was. And when I started this project, I started with the obvious experiment of just determining if Marlin is required for pancreatic morphogenesis. And Marlin is encoded by this gene called NF2. So we obtained NF2 flox mice. We crossed them to a pancreas-specific pre driver called PDX-pre and generated Marlin pancreas-specific loss function mice called NF2-PancKO. And so for the rest of this talk, I'll call this Marlin pancreas deletion NF2-PancKO. And I just wanted to see if at first if the mice survived and they had any pancreatic defects. And so these are adult pancreases from 3 to 4 month old mice. The mice do survive, but they have a few different problems. One of the problems they have is just a very dramatic reduction in pancreatic mass. So the pancreas has been outlined in this light blue color right here, and you can see the stomach and the spleen. And the pancreas is only about 1/4 as big as it should be in these mutants. And we took a little bit more look at what the morphology of these pancreases were like. So this is IAF of hormone IF of some adult pancreata, stained from mucin-1 to show you all the fine lumens, and DBA, which shows you the ductal lumens. And you can see at this stage we have this sort of nice ductal tree, and the septal tree extends into these fine mucin-1 positive tip lumens. In the knockouts, however, you can just visually see that they're pretty dysmorphic. So instead of fine mucin-1 positive lumens here, you instead have these sort of dilated sort of bag-like lumens here. And instead of a ductal tree, you kind of just have this really dysmorphic ductal structure instead. So we wanted to also assess endocrine formation and function because endocrine is so important for disease-related things. And so we look for endocrine markers for beta cells, insulin, and alpha cells, which is glucagon. And in mice, the mature islet has this really nice mantled structure where the insulin positive cells are in the middle, and then the glucagon positive cells sort of surround the outside. In the NF2 mutants, we instead get this commingling structure here, and you can see that reflected in the quantification that not only are the islets malformed, but they're also somewhat significantly smaller. We also assessed endocrine function by doing a glucose tolerance test. This is an assay where you challenge the mice with the bolus of glucose and essentially watch how fast the mice is able to clear that glucose from its blood levels. And the mutant mice had defects in this assay as well. So all suggestive that myelin is really required for both endocrine function and formation. Okay, and then we wanted to understand really what was going wrong to create this phenotype. And I think to really understand when something goes wrong, we have to know when your protein deletion is happening. And so this is again driven by PDX1 Cre. In mice embryos, PDX Cre is starting to be expressed as early as E8.5, but previous studies have reported that deletion occurs between 10 and 11. So we wanted to see if that is true for us. And so this is control of mutant tissue stained for myelin and mucin-1 at E10.5. And you can see at 10.5, so in the control, there's myelin really nicely along these mucin-1 positive membranes. And the mutant, that is true as well. So it doesn't look like deletion is really complete. But by 11.5, we get a pretty nice deletion of myelin, which is in magenta from the apical membrane as identified by mucin-1 staining, which is in here, cyan. So then we started to look, so then we concluded that deletion starts at approximately 11.5 in these mutants, and we started to look a little bit closely at the phenotype. So we did some tracing of the phenotype of 11, 12, and 14. And at 11, and I won't show you all this data just for the sake of time. At 11, there's not much of a phenotype. By 12, we can start to see it, but by 14, it's really robust. So I'm going to show you the 14.5 data. And so this is a whole mount of an E14.5 pancreas stained for mucin-1 to show you that luminal network. And so you can see kind of in the background here, the plexus lumens, and then they end in these tip regions at the outside. In the myelin mutants, instead of a nice plexus, you instead get these bag-like lumens. They still end in tips, but you can just visually see that there's fewer of them. And if we look at the quantification, we can see that the plexus complexity and the number of tips are reduced. So really showing you that there's a problem with the formation of this luminal network. We wanted to take a little bit of a closer look at what these bag-like lumens that we see towards the middle of the pancreas were. So if we did some really high resolution microscopy, and what we see is that these bag-like lumens are regions of stratified epithelium. So shown here is an optical cross-section taken somewhere from this max intensity projection, where we've stained the pancreas for ECAD in cyan, mucin-1 in yellow, and laminin in pink. And you can see that in the control, there's pretty much one cell layer between the apical membrane and the basal membrane, suggesting that we've successfully transitioned into this monolayer tree state. In the mutant, however, instead of one cell layer, we have anywhere from 4 to 5 cell layers, showing that we're still in a stratified cell state. And so we wanted to better understand, how the stratification might be being mediated, because in pancreas, stratification is sort of linked towards this formation of this luminal network. We thought maybe there was an issue with apical polarity. So we did some stains for canonical apical polarity things, one of which is this tight junction marker called ZO1. And so ZO1 is really strongly apically expressed in the control epithelium, so you can see it really nicely. In the mutants, where we have a lumen, we still see ZO1 being apically expressed. But if we look, for example, at these cells over here, even in the XZ position, we actually can't detect ZO1 at all. And these cells also end up lacking mucin-1, suggesting that these cells may not be able to participate in lumen formation. As another marker to check how these cells are polarizing, we looked at Golgi. So in control cells, Golgi are localized just right below the apical surface. So you can see that right here. The Golgi has been stained with GM130, which is shown here in pink. And then in NF2 PinkKO, where we have a lumen, we similarly see the Golgi to be localized right underneath it. But where we have these regions of stratified epithelium, we start to see that these Golgi seem to polarize towards each other, but are unproductively able to make a lumen. So what we think may be happening is that we have these first initial lumens that form in the presence of Merlin because our Cree is not quite deleting it yet, but we're not able to induce the formation of the next lumens. And so we wanted to use another system to look at this. And so we wanted to really be able to like live visualize that process. So we started to turn to an explant system. So what we do is we take the pancreas out of the mice at about E11.5. We plate it on some fibronectin, and then we're able to sort of watch it grow. And so this is just a pancreas and the control pancreas, and you can see that at 72. But by the end of it, we have this really nice stratification phenotype. So showing you that the explants recapitulate what's going on in the in vivo mutants. And you can see that reflected in the quantification as well. And we can use the same system for live imaging. So this is just the control video. And what I hope you can appreciate is that we get this sort of nice coordinated growth. And we'll additionally get the formation of these new branch lumens called polarized canals. So just watch these regions with the white boxes and you'll start to see some form. In the mutants, however, this process doesn't really happen. Instead, we get continual growth of the existing pancreatic lumens. So, and you can see that also reflected in this quantification as well. And so we wanted to test more directly if Merlin was required for lumen formation, but again, because of the timing of our pre-deletion, that was a little hard to do in vivo. So we turned to an ex vivo system where we take the pancreas at E12.5, long after Merlin has been completely gone. We dissociate them into single cells, and then we culture them in matrigel, and then we can actually see a little sphere that has a lumen form. And so we looked and we did this assay, and in the control cells, you can clearly see this luminal space between cells. But in the mutant, that luminal space just doesn't really form. So we think that Merlin is required to coordinate lumen formation. So how are new lumens made? So there's been a lot of work on this from our lab and others, which shows that you really need polarized vesicular trafficking to rearrange junctions and deliver apical materials to the apical membrane of the developing lumen. And using the same system we've developed before, just at a higher resolution and a higher frame rate, we can actually watch this process happen in real time, where these little yellow dots here are trafficking to our vesicles that are being trafficked to a developing lumen. So we know that polarized vesicular trafficking is really required. And we looked in our control of mutants to see if potentially some of these key vesicular trafficking effectors are impacted. So shown here is IF for RAV5. You can see there's a really nice apical enrichment of RAV5. But in NF2 mutants, there's not only an increase in RAV5, but also instead of being just apically enriched, we get sort of a basolateral mislocalization. And we see a similar pattern for CDC42. So we think that Merlin is important for vesicular trafficking control. And we wanted to better understand the molecular mechanism behind this process. So we did some single cell sequencing, and this is the gene ontology analysis from our single cell. We were really relieved when hippo signaling came back up because, I told you we got into this for hippo signaling. And so we performed some IF to validate that hippo signaling is indeed up, and you can see a pretty nice increase in nuclear YAP and a decrease in the inactive form of phospho-YAP in my mutants. And so we decided to try to breed in some YAP and TAS flox alleles to create a YAP and TAS haploinsufficiency model. And so shown here are whole mounts from control and NF2 mutant YAP TAS haploinsufficiency mice. And you can see that the luminal plexus doesn't look really all that much different from NF2 mutants. And if we look at some laminin and mucin 1 staining, you also can see that the destratification phenotype didn't really get rescued. So it didn't seem that Merlin's primary mode of action for this particular part of the phenotype was through YAP and TAS. So we went back to our single cell to see what else might be implicated. And we noted PI3K signaling terms were also elevated. And this actually caught my eye because there's been some really nice papers about PI3K signaling in the pancreas. And so this is a P10 knockout, and the P10 is a condition which overactivates PI3K. And it has a lot of similarities to my postnatal NF2 phenotype. So in both the knockout of P10 and my mutant condition, you can see the development of these sort of cyst-like conditions, and then there's a loss of these acini that we see at the periphery. And so I thought potentially Merlin could be working through PI3K given the similarities in these phenotypes. And so PI3K pathway is a pathway that's governed by tyrosine kinase receptors, which leads to the activation of PI3K, which is opposed by P10, and then leads to a variety of downstream signaling. And so one thing you might expect if PI3K is high is, awesome, thank you, an increase in phospho-AKT. So we turned to a cell culture situation to see if we could see that. And so this is pancreatic ductal cells that have been treated with either non-targeting siRNA or Merlin siRNA. And you can see a really nice increase in PI-AKT phosphorylation in the NF2 knockdown conditions. And if we treat with a drug that inhibits P10 or overactivates PI3K, we get a replication of this stratification phenotype. And so we wanted to also directly test if inhibition of PI3K could inhibit, it kind of ameliorate the Merlin phenotype. So we used a drug called LY2-94002, which directly inhibits PI3K, and we treated our explants with this system. So in the control, there wasn't really much of a difference between VMSO and drug-treated pancreases, but if we look at our mutants, what we see is that towards the tips of these explants, we actually do see some level of rescue. So it's not a complete rescue. We still have these regions of stratification, but if we look closely, we can see that there's more of these polarized tips polarized canals that are forming. And so just to conclude, what I think I've shown you so far today is that in the control embryos, we have this initiation of lumen formation. So as a lumen is developing, it will induce cells near it to form these polarized canals. These polarized canals are formed through vesicular trafficking. And then as that process occurs, you get this really nice coordinated luminal growth. In the Merlin mutants, however, this process really goes awry. So instead of inducing additional lumens, we just continue to kind of grow the lumens that are there prior to complete deletion of NF2. And eventually we get these dysmorphic bag-like lumens, which we think are driven in part by hyper-elevated PI3K signaling. So with that, I'd really like to thank my mentor, Dean Cleaver, and all of our collaborators and everyone who's helped me along the way. And I'd also like to thank my funding sources for letting me continue to do this kind of work. And with that, I'll take any questions.
Thank you, Neha, for the excellent presentation. So I would like to remind our audience, so you can submit your questions through the Zoom Q&A box. So while you guys are preparing for the questions, I will begin with mine, I guess. So I really like the data you showed about, PI3K inhibition kind of rescue the phenotype, right? So my question is, so since, PI3K also controls proliferation, so have you checked about, anything to do with cell death, proliferation,
Yes.
things like that?
Yeah, so I've checked really extensively proliferation and cell death, and embryonically, there's not much of a change in either proliferation and cell death. At some of the postnatal stages, we start to see increases in cell death. So I think that is definitely a thing that's happening towards the later stages, but I don't think that's what's causing the phenotype to go wrong in the early mutants. Does that make
I
sense?
see. Yeah, I think that totally makes sense. So actually another question. So, I'm interested in like, have you tried to, right, because you have those, complicated 3D structures, right? Like in vivo, sometimes it's hard to quantify it or like, characterize it because, depends on where you image or like the structure could be different, things like that. So have you tried to expand those cells or like tissues into a dish, like to check if they have any phenotyping, maybe migration, like, things like that? Yeah.
So like I've cultured the cells just flat. I will say pancreatic progenitors are sort of notoriously finicky. And so that you can't passive them for more than 2 or 3 times. But they don't look like by and of themselves, they don't look that different cultured flat than the mutants. It would be fun to do something like a migration assay on those, and that's in part why we've been doing stuff with SIs in a cell line, but also those are immortalized, so they're also there's challenges.
I see. Yeah, I see all the problems here. Yeah, so I guess I have some questions. So first, we have a question from Michael. So had in chat box. So had Merlin been implicated in the PI3K regulation prior to this study? So any speculation about the mechanism at this point?
Yeah, so there is a couple studies that have implicated Merlin in PI3K regulation in the context of like neural tissues. So in brain tissue, there is a molecule called pike-L, which Merlin directly interacts with, and pike-L then works to modulate PI3K. But that isoform does not exist in pancreas. At least I cannot detect it either by like RT-PCR or it's not in my single cell. So it could be a novel enhancer or novel protein. Another thing that we've been speculating is that Merlin has been kind of implicated in receptor, in like maintaining the ability of having concentrations of receptors at the membrane. And there's 2 PI3K tyrosine receptors that we've been interested in. So we might think that Merlin's function is to stabilize those receptors at the membrane. Loss of Merlin might lead to internalization of those receptors, which then potentiates PI3K. So that's broadly what we're thinking. But the data is coming.
Mm-hmm. So, and I got another question from the Q&A box. So from Blancha, so how do you think the Golgi plays into this phenotype?
Yeah, so if you look at the E10 or E11 pancreas, which is still sort of a stratified epithelium, where there's not a lumen, the Golgi will sort of be in this random orientation. And so we think we kind of get a predurance of that state because as the cells polarize, they rearrange their Golgi to be closer to the apical surface. And so what I think is happening is that the cells that have a kind of a stratified epithelium with the or non-polarized Golgi are just basically haven't been able to go through the polarization process. Does that make sense?
Yeah, I think so. So I think there's no more question, but I
have a question in the box.
Oh, yes. Sorry. Yeah, I see that. So like Marlin potentially have functions in cell polarity in addition to having functions in the hippo pathway. So your phenotype looks like classic cell polarity defeats. Have you thought of knocking out crumbs to tell these options apart?
I think that is a good point. We haven't knocked out crumbs. We have knocked out a couple other proteins involved in sort of establishing polarity, including ephadin and RAB11. And so one of the things that has been kind of tricky to tease apart is often the ones where we have completely ablated polarity, we end up with these sort of abortive microlumens which are disconnected. I don't quite see disconnected lumens. It's almost like the lumen I'm getting is this hyperconnected phenotype situation. And so I think it is an important point that, we do have a cell polarity molecule that seems to be going wrong. But I'm not sure if it's the same phenotype as we would get if we knocked out Prums.
So I guess this time we don't have any more questions. So I think, thank you. I would like to thank to our today's speaker, Dr. Gopal Kushawah and Dr. Neha Ahuja, for your excellent talks. So this seminar has been recorded and will be available on the SDB website next week. So please join us for next month's seminar on Friday, March 13, when Dr. Martha Echevarria Andino from University of Wisconsin-Madison and Dr. Fernando Palominos from University of California, Berkeley will present. Thank you all for coming.